Good afternoon, everyone. We'll start today's session with Ganesh Vandana.
वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
Welcome to the ninth day of Series 1 Online Faculty Development Program on Emerging Trends in Drug Design and Development, sponsored by AICTE New Delhi and organized by Roland Institute of Pharmaceutical Science, Barampur. Today we have with us Dr. Malay Rana Sir, presently working as an assistant professor in ISR Barampur. Prior to his talk, I take this opportunity to introduce Sir to all of us. Dr. Malay Kumar Rana sir has obtained his bachelor's in chemistry from Vidyasagar University in the year 2002 and master's in chemistry from Vidyasagar University in the year 2004. He then joined IIT Kanpur for PhD in physical chemistry under the supervision of Professor Amalendu Chandra sir. After the completion of his PhD in 2010, he worked with two research groups as a postdoctoral fellow, namely with Professor Marco Massia, University of Sassari, Italy, from the year 2010 to 2011, and Professor Donald J. Segal, University of Michigan, USA, from the year 2011 to 2014. Subsequently, having spent almost a year in IIT Kanpur as a senior scientific officer in the group of Professor Siddharth Ponda, he joined ISR Barampur in 2016, initially as a visiting assistant professor and later in 2017 as assistant professor in Department of Chemical Sciences. His main areas of interest are solvation of non-polar solid materials, gas separation and storage, including carbon dioxide and methane gases, therapeutic discovery of critical illness such as coronavirus, cancer, etc. With this, I request Sir to come on board and continue with today's session. Sir, you can continue, sir.
Yeah, thank you, Jasma, very much for your kind introduction. At the onset, I would like to thank the principal, Professor Ravi Kumar, as well as co-coordinator, Professor Vishwa Mohan Sahu, for their warm invitation. I am very much glad to participate in this faculty development program. I would talk on a very recent problem, which is coronavirus, and how we have put our efforts to make some discovery of effective drug molecules. So please allow me to let me share my screen. Okay.
Sir, please make it full screen.
Yeah.
Yes, sir. Very clear, sir.
Okay. Thank you once again for this invitation. So I would talk on a very recent problem, as I mentioned. This is basically on COVID-19 infection and therapeutic discovery. So I am Dr. Malay Kumar Rana, Assistant Professor in the Department of Chemical Sciences, ISR Barampur. So, in the last 1 year, we have been through the worst situation, worst period. It is the worst situation in the century. This is caused by severe. Okay, so this situation arises because of the severe acute respiratory syndrome coronavirus 2, in brief SARS-CoV-2, which caused COVID-19 infection. So coronavirus or SARS-CoV-2 is a new virus which resulted an ongoing pandemic, uh, probably the biggest pandemic in the century. So till today, there are 112 million confirmed cases of COVID-19 infection, out of which 2.49 death cases are reported. Also, uh, there are 63.4 million recovery cases. The first coronavirus infection was reported in Wuhan, Hubei province of China in late 2019, when it was noticed some unidentified pneumonia in some people. It was thought that bats are the natural hosts of SARS-CoV-2 virus. So the virus is originated from bats probably, and then it gets transmitted to humans through the intermediate hosts like pangolin and snakes. It causes respiratory tract infection. When a coronavirus infects humans, basically it infects first the respiratory tract. In the in the mild infection, basically the upper part of the respiratory tract is infected, such as sinuses, nose, and throat. In the severe infection, the lower part of the respiratory tract uh, gets infected. So this is shown in this picture. Basically, it captures the entire respiratory tract, causing problem in breathing. Now, the key events in the SARS-CoV-2 outbreak, it started, or the first outbreak takes place on December 30, 2019 in Wuhan city in China. And then on 7th January, the virus was isolated, and on 12th January, the sequence of the virus was analyzed. Now, from the sequence analysis, it was told that bats uh, may be the potential origin of this virus on 21st January. However, this statement that bats are the potential origins of this SARS-CoV-2 virus, it's debatable, because whenever the sequence comparison of the SARS-CoV-2 or sequence and bat coronavirus sequence was analyzed, it was found that there is a similarity of 96.2%. Okay, not 100%. So it means that whatever the virus, this SARS-CoV-2 sequence, okay, that is not the original sequence of bat coronavirus. So bat cannot be the potential origin of the virus, but uh, SARS-CoV-2 has been originated from the genetic engineering of the bats coronavirus, probably maybe in some labs. So anyway, so when the first outbreak takes place on December 30 in some people, about 27 people came across having unidentified pneumonia, and in a week this number got doubled. Okay, and then In third weeks, around 21st or 23rd January, um, this infection case has reached almost 6,000. So there is a big jump. Looking at the situation, uncontrolled situation, the World Health Organization on January 30, they declared public health emergency, and they named this new virus as SARS-CoV-2 or COVID-19. And following up this incident, the more studies, investigation about this virus, how it infects, have taken place, and people became cautious, and the pandemic spread from one corner of the globe to the other corner of the globe. So this is basically a rough milestones of the events that how I mean, so when it originated and how it spreads. So here, next, we will talk about that how this virus infects or it spreads. Okay, uh, whenever the virus is originated in China, then people lived in China, especially the people from other countries. So they were allowed to go to their native countries, and those people were infected by the virus. And whenever they reach their native countries, the people in the native countries, they got infected due to human to human transmission. So the spread occurs because in a way that whenever the infected person talks, he sneezes or he coughs, then droplets are created, and the droplets can travel as long as 6 feet distance. Whenever the persons surrounding the infected person, um, they receive these droplets in their nose, eyes, or mouth. Um, when they inhale these droplets, they get infected by the virus. Not only that, whenever these droplets are settled down on some surfaces, and a healthy person comes in contact with those surfaces unknowingly, they come in contact with such droplets, and ultimately the droplets go inside the body through nose, eyes, and mouth. Now, the question is that whether after a long gap, after many hours, whether these droplets are still capable of infecting the person, the coronavirus is active in the droplet or not. Here, the study show that in aerosol, basically in suspended dust particle in the air, the virus can live up to 3 hours. On the copper surface, the time period of lifetime of the virus is 4 hours. On paper surface, cardboard, it can live up to 24 hours. On plastic or stainless steel surfaces, the virus can survive up to 2 to 3 days. So, whenever these droplets are settling down on some surfaces, still there is a risk that the healthy person can get infection by touching those surfaces. So sometimes the infection is not visible, means the symptoms are not visible. These are asymptotic. Sometimes this so coronavirus symptoms are asymptotic in nature. What are the most common symptoms? Whenever we are infected by coronavirus, the most common symptoms are basically fever, there is chilling, fatigue, cough, sore throat, shortness in breath, and loss of smell and taste. These are the very common symptoms of coronavirus infection. The symptom may start in 4 5 days, or it can start as a later time within 2 weeks time period. So, as I said earlier, the some people may have symptoms immediately. Some people may not have symptoms, although they are infected by the virus. So, this virus is basically very very mysterious in nature. Uh, There are some least common symptoms also seen, such as headache, muscle aches, diarrhea, show abnormal heart rhythm and heart failure, and stroke. Basically, reports say that reports point out to multi-organ failure in COVID-19 infection, septic shock, blood clots, or long-term damage of organs. So these severe accidents or incidents uh, caused by the coronavirus infection have been also reported. Many patients recovered fully in about 2 weeks period, and in other instances, the symptoms like breathlessness and fatigue may persist after 2 weeks also. Now, still, I mean, although we have heard of, we have come to the stage of where basically vaccine has been in the market, and many people have been vaccinated nowadays, but still, we have the news of recent strains of coronavirus. We are not totally immune to the new strains of the coronavirus. Still, we have to follow this some preventive measures like frequent hand washing, social distancing, quarantine. So whenever we are talking, coughing, etc., always we should cover our mouth, face, and it is instructed to use N95 category of marks. So till now we have to follow the preventive measures. Although there are some remedies in the market, but still we are not totally immune to. We have not achieved that kind of immunity which is totally able to fight against the all sorts of coronaviruses, particularly the new strains of coronavirus SARS-CoV-2. Now, what are the risk factors? Uh, it has been anybody can get coronavirus. In the most infections are mild, okay, and the older people are at a higher risk. It has been found out that people younger than 50 years old, uh, the death rate is less than 0.5%. On the other hand, people who are older than 70 years, for them, the survey tells the death rate about more than 8%. So basically, aged peoples are are at a higher risk in case of the COVID-19 infection. And it has been also found out that pre-existing health conditions, especially if somebody has diabetes or heart disease or BP, hypertension, etc., uh, so they are more likely to succumb to the virus. Most of those who died of COVID-19, it has been found out that they had some pre-existing health conditions, mostly either cardiovascular disease or diabetes or hypertension. So not only these diseases, there are some other diseases which can make a person vulnerable, or which can aggravate the COVID-19 infection in a person. So these are as follows. These are basically, if you have kidney problem, you have obesity problem, if you have pulmonary diseases, if you have asthma, if you have liver diseases, okay, so these sorts of health issues can aggravate the COVID-19 infection, and the person can be at a higher risk of losing his life. Now, so whatever till now we discussed about the nature of the infection. Now we will try to understand here that what is coronavirus structurally, how it is built with, and what components of the coronavirus is going to infect us, how the infection takes place in human body. So, coronavirus, the picture of coronavirus is like this. So it has a globulous shape, and there are some crowns, red crowns onto it. So these crowns or club-shaped structures, they are erected out of the surface. And the average diameter of the coronavirus, this globe, is of 120 nanometer. However, the size of the coronavirus may vary. It may be smaller than 120 nanometer, or it may be higher than 120 nanometer. Usually, if we take into account the extreme sizes, the coronavirus may vary in size from 50 nanometer to 200 nanometer. So there are several structural components, as one can see in the picture. Whatever the cover, this gray cover, whatever we can see, this is nothing but the lipid bilayer, which covers up the genetic material RNA. This is coronavirus is basically having a single stranded positive sense RNA. Now, on the surface, the cloud-shaped structure, red color structure, it looks like a crown. It has a primary head. You can see the upper part. There are three heads here. It is clearly visible. And if somebody knows cards, so in the card, this structure is basically a club like structure. So it actually this club shaped or the it looks like a crown. So this structure or whatever the structural component, this is known as the spike protein. So this is spike protein. Uh, Now, whenever the coronavirus is it's looked through the electron micrograph instrument, because of these red globe-shaped structures, uh, the whole coronavirus looks like the corona of sun, and that's why the name coronavirus has been given, because in the electron micrograph instrument, coronavirus looks like the corona of the sun, and that's why it is called coronavirus. So anyway, so this is the bigger structure, about 20 nanometer long, erected out of the surface. Apart from that, similar to this, okay, there is another structural component which is white in color, shown here. It almost similar. It is almost similar in looks like the spike protein. This is called hemoglobin. Uh, this I will tell that what are their functions, and then after hemoglobin, there are two more protein structures which are shown in two different colors. So this one and the other one which is shown in yellow color. So this is called the membrane protein, and the yellow one is called the envelope protein. So After this, the RNA basically is covered with a protein which is called nucleocapsid protein. So this is the RNA. So on surface of it, it is covered with some protein which is called nucleocapsid protein. So here, all these structural components, they have certainly some work. The spike protein basically helps in the attachment. Here it helps the virus to get attached to the human cell, and thereby it mediates the infection. The hemagglutinin, this looks like the spike protein. It helps the attachment and detachment of the virus from the human cell surface. So while the virus is attacking the human cell, then it assists the attachment process, and later on, whenever new viruses, say once the infection is initiated in human cell, and thereby replication takes place and new viruses are reproduced inside the human cell, then they come out of the cell, come out of the cell, and in that incident, the semaglutine also helps in the detachment of the new viruses from the cell surface. Now, envelope protein or membrane protein, basically they hold up this lipid structure to maintain the shape of the globular shape of the whole virus structure. So the functions are stated in this table, but I did not tell about the positive sense single-stranded RNA. As you know, that RNA is the genetic material, and okay, it it basically have some transcription to synthesize viral messenger RNA as well as replication of the genome. Basically, it replicates okay, to reproduce new viruses through producing messenger RNA. The whole genome is given here. The coronavirus genome is almost 33,000 uh, kilobases. Uh, there are several regions. It has open trading frame 1A, it has open trading frame 1B, and there is also open trading frame 1AB. Open trading frame 1A and 1B, they cover almost two-thirds of the whole virus genome. Okay, and they encode several non-structural proteins. So earlier, whatever we discussed, these are the structural proteins. They have particular structure you can see, but in the viral genome, whatever the open trading frame 1A and 1B, they are encoding they are encoding the non-structural proteins. The proteins which are basically expressed when the virus goes inside the cell, okay, and takes part in the viral the genetic material RNA. It undergoes translation and replication. That time, these protein parts expressed inside the cell. So that's why okay, these proteins which are encoded by these two regions are called non-structural proteins. They are usually they are non-existent, but whenever the virus gets inside the cell, okay, these proteins appear inside the cell. Uh, the other reading frame, which is open reading frame 1AB, that represents the structural proteins like okay, membrane protein, nucleocapsid protein, spike protein, etc. Now here we will discuss a little bit about how the virus infection takes place. We have the coronavirus. Coronavirus comes closer to the human cell, basically lung cells. Okay, and Coronavirus has the spike protein. We mentioned that spike protein has a role. It helps in the attachment of the virus to the cell surface. So there are receptors in lung cells, and these receptors are called angiotensin converting enzyme 2, in brief ACE2. So there are ACE2 receptors in the human lung cells. Okay, coronavirus come, and with the help of spike protein, they get attached to the ACE2 receptor. And what happens after the attachment? The membrane fusion will take place between the coronavirus and human cell, and the virus gets inside the cell. Um, After that, the genetic material is released inside the cell, and there will be translation, replication. um, uh, then during the translation, the non-structural proteins are produced, and followed by the production of messenger RNAs. Now every RNA, they collect the other structural components, basically the other structural proteins like membrane protein, nucleocapsid protein, spike protein, to build the new replica of the coronavirus. So they basically these mRNAs are getting converted into the new virus. The new virus is covered with the vesicle coming from Golgi. So these vesicles help to traverse the newly formed virus from the cell inside to the cell surface, ultimately with the help of hemagglutinin or with some mechanism, the new viruses get out of the cell. So, in this way, the infection takes place once one cell is infected. The new viruses reproduced in that infected cell go to the other cell and invade a healthy cell, and thereby again it infects and kills the cell. Again, the reproduction of new viruses will take place, and that's the way the infection spread. Now, with this brief introduction about the structure or the mechanism of infection of the coronavirus, here we have attempted to uh, discover or design some therapeutic which can fight against the virus. So our design strategy it's based on three aspects. One is design of biomimetic peptide. This basically designing peptides which can interact with the spike protein, and thereby it can prevent the attachment of the spike protein to the ACE2 of the cell surface. So thereby, if it can stop the attachment of the spike protein, then it can stop the virus infection. So so one strategy is to inhibit the virus at the entry point. The second strategy is that without designing new molecules or new peptide molecules, whether we can screen out the existing drug molecules available in the market to know that among them which are going to inhibit the virus, which is called basically drug repurposing, because all these drugs, whatever in the market, they were launched against a particular disease, but we don't know that apart from that particular disease, the same drug whether can be able to act against other diseases or not. Since these drugs are available on the market, so the shortest route is to screen them, is to test them against the coronavirus, to know that whether some of them are going to act against the coronavirus or not. So this is called drug repurposing. In the drug repurposing, we basically may consider some groups of drugs against the main protease. So it is the protease molecule in the virus, one of the protein targets in the virus, which performs a very vital role in the infection process. So we try to screen out the some existing drugs against this particular protease target, the main protease. The third strategy was target identification. So, after the outbreak, maybe 2 months later, after the outbreak of the virus, there were some medicines like ivermectin and famotidine were reported to have positive effects on the COVID-19 patients. But how they are acting against the virus, what is their mode of action, it was not known. So this interested us to consider them, to find out that on which protein target, or on which portion of the virus they are going to attack the virus. If we can know that okay, they are targeting these protein targets of the virus, then targeting those protein targets, one can design better drugs to prevent the virus infection. So that was our another strategy, just to give some binding insights into the interaction of ivermectin and famotidine with the virus molecule. Uh, Lastly, based on these studies, we proposed a combination therapy which can offer a better treatment of COVID-19 patients, which I will discuss at the end. The methodology we usually adopted computational methodology. It is like this: you get the target protein of the virus, you get the drug molecule, and we dog the drug molecule or inhibitor molecule with the suitable target of the virus. Uh, so if we have several drug molecules in hand, or several target molecules, then we perform docking, and we collect the docking scores. Based on docking scores, we screen the drug molecules as well as target molecules, that which are the best combinations. And now the best combination molecules, or the best complexes, drug protein complexes, those are subjected to further molecular dynamic simulation. In the molecular dynamic simulation, after the simulation, we perform certain analysis, such as analyzing the structure of the protein complex captured by root mean square deviation, root mean square fluctuation, radius of gyration, principal component analysis. We can again compute the binding energy of protein target, which is called free energy calculation here. So basically, post molecular dynamics analysis helps to confirm the screening, that whether you have correctly screened out the drug molecules or the protein targets or not. The molecular dynamic, post molecular dynamic analysis will tell you that whatever the selection you have made, that is the right selection or not. Now, coming to the first one, biomimetic peptide design. So this is the complex structure of spike protein bound to ACE2 protein, human ACE2 protein. This is the crystal structure collected from the patient. So in this crystal structure, one can see that this is the receptor binding domain of the spike protein. Okay, and this receptor binding domain region is interacting with the helix of the ACE2 protein, and this helix is called alpha 1 helix. In the picture on the right hand side, it is shown that this is the human lung cell. You can imagine, and from the lung cell, in the lung cell surface, there are ACE2. It has the receptor binding domain. This receptor binding domain is interacting with the S1 domain of the spike protein, and this is S1 domain. Basically, it comprises the primary head. I pointed out earlier that the spike protein has the primary heads, which look like the club. So RBD is going to interact with H1. Then what happens? Due to interaction, the H1 domain gets attached from the H2 domain. Now, the H2 domain or the rest of the virus, with the help of transmembrane protein, it combines with it gets fused with the membrane of the cell, and thereby it gets inside the cell. So this is the mechanism of infection. Now, this alpha 1 helix, we have given here that what of the amino acids present in the alpha 1 helix. So here we can, I want to stress upon one thing that compared to SARS-CoV, okay, SARS-CoV-2 is manifold infectious than the earlier SARS-CoV. The reason is due to mutation in its sequence. So we have you can see here that we have performed the sequence similarity study, and one can see that the white regions where basically the amino acids sequences of two viruses, they are not matching. That means in these white regions, the amino acids of the earlier SARS-CoV have been mutated. And because of this mutation, the interaction of the spike protein with the spike protein with the ACE2 is manifold stronger than the spike protein of the earlier SARS-CoV. So this is basically attributed to the mutation in certain regions of the old SARS-CoV. Now, in our studies, computational studies, we found out that which residues in the alpha 1 helix they are interacting with the RBD receptor binding domain of the receptor binding in domain of the spike of the ACE2 cell. Now, since alpha 1 helix is interacting with the spike protein, therefore one can think of at the easiest way that if we just take only, if we make a therapeutic, which is nothing but alpha 1 helix, then alpha 1 helix, the peptide medicine, it can compete with the alpha 1 helix of the ACE2 to interact with spike protein. So spike protein then can get attached to that alpha 1 helix peptide, which has been customized, and it will not actually interact with the lung ACE2 alpha 1 helix. But since okay, the structurally this alpha 1 helix in the ACE2 of lung, it's the same as the is the same as the alpha 1 helix you have made, then still there is a possibility that spike protein will go to the ACE2 and interact with alpha 1 helix. Our rationale is that if we can just look up the amino acid which are not interacting with the spike protein, and if we mutate those amino acids to enhance the interaction of this alpha 1 helix segment with the spike protein, then whatever the peptide inhibitor we will design, certainly they will have the stronger interaction with the spike protein, and they can be able to divert the spike protein to interact with them rather than to interact with the alpha 1 protein, alpha 1 helix of the AC2 protein of lung. So this is the strategy. We start with then alpha 1 helix. We looked at which are the non-interacting residues. We gradually mutate these non-interacting residues to make a peptide library. We ultimately made 124 peptide molecules from alpha 1 helix. Now we analyze the interaction of the wild alpha 1 helix and the peptide molecules whichever we have derived with the spike protein, and we screened out those peptide molecules which have larger interaction or stronger interaction than the wild alpha 1 helix. So that way we try to reach to some biomimetic peptide inhibitors which can be used as therapeutic molecules. So here, so these are the, I am showing here the top 13 designed peptide molecules. So these molecules, so this is the sequence of the original alpha 1 helix, and now you can see here in all other peptide molecules we have mutated certain residues. For example, in peptide 1 we have mutated E to D. Okay, and then in peptide 2 we have mutated E to or W. So these letters representing different amino acids. So we have performed single point mutation to derive a peptide library. We have performed also simultaneous mutation. So in the top 13 list, whatever I am presenting here, the first 12 molecules, they are originated out of single point mutation, and the last molecule, the peptide 13, it has been originated by taking into consideration of the simultaneous mutation of all non-interacting residues of alpha 1 helix. And now you can see that what is the binding interaction. In case of alpha 1 helix, the binding energy is a minus 118 kilocalorie per mole. The base peptide given at the end, it has the binding interaction energy is minus 150 kilocalorie per mole. There are several other molecules which have more negative binding energy than the alpha 1 helix. The more negative binding energy compared to that of alpha 1 helix, it means that a stronger interaction between the peptide molecule and the spike protein. So, so this is these are the values coming from the initial docking results. Now, whatever according to the docking energies, we have shortlisted the molecules, and these are the 13 shortlisted molecules I showed here. These are considered for further molecular dynamics study, which will basically tell you that which peptide molecules, when complexed with the spike protein, having the would have the more stable complex compared to the alpha 1 helix and spike protein complex. So the values, the stability parameters judged by RMSD, RMSF, RG, and also binding energies, these are provided here. Basically, if a complex has a higher value of RMSD, RMSF, RG, okay, that means the complex is less stable compared to the other complex. You can see, so these two columns are for spike protein and alpha 1 complex. This alpha 1 basically the wild alpha 1 peptide molecule, and these two columns are for the base peptide inhibitor. You can see that RMSD, which is represented by the pink column, the RMSD value of the base peptide inhibitor 13 is lower than the RMSD of alpha 1 helix. So it means that the inhibitor 13 and spike protein complex is more stable than RMSD. And in terms of the binding energy, you can see that binding energy means the free energy. You can see that for the peptide 13 inhibitor, we have more negative binding energy. That means we have more stable complex compared to the alpha 1 helix. Almost we have 3 times more negative binding energy for the base peptide inhibitor 13 compared to alpha 1 helix. So basically, whatever conclusion can be made out of these studies, the similar conclusion can be drawn from the study of the radius of gyration. Here in this plot, I just want to highlight one fact that whenever we are going to say mutate, I told you that the logic behind the design of peptide library is to mutate the non-interacting residues present in the alpha 1 helix. So whenever we are mutating, say lysine to tryptophan, then you can see the contribution of the lysine was positive in the alpha 1 helix and spike interaction, but now it has the negative contribution, negative energy. So that way, the uh, design peptides have the better interaction with the spike protein than the alpha 1 helix. This is also true whenever you can see that when asparagine in 33 is mutated to aspartic acid, D33, then the interaction energy or energy contribution becomes negative. So the picture below, you can see lots of interactions present between the base peptide inhibitor and the spike protein. So this is the base peptide inhibitor and spike protein. Again, here we have basically overlapped the initial and final structures of the complexes. We have considered 2 complexes. One is alpha 1 helix and spike protein complex, and which is given in the upper panel here, and the other one is the base peptide inhibitor 13 and the spike protein complex. So initial configuration means before the molecular dynamics, the complex structure was is presented in green color, and after dynamics it is the cyan color. You can see basically there is a structural or conformational difference before and after the molecular dynamics. So this difference indicates that the complex or the binding between alpha 1 helix and spike protein is not that that great. It's not that strong. But whenever you looked into this complex, the initial and final structures, you can see a very small deviation, not much. Okay, so here you can see a large deviation of the alpha 1 helix from its initial position. But here alpha 1 helix, okay, before and after the dynamics, they remain in the same location almost, or also for the spike protein. So from this visualization, it is clarified that whatever peptide or the base peptide we are claiming, that has certainly a stronger interaction than alpha 1 helix. So if we use this molecule, peptide molecule, as a therapeutic, then it is, according to our study, certainly going to inhibit the virus entry. It can prevent the attachment of the spike protein with the human lungs. So here pictures on the right, here we have just shown the designed peptide complexes with spike protein of the 13 peptides we have designed. Now, I will come to the topic of drug repurposing. So I told you the purpose of drug repurposing is to is to quickly catch those promising drug molecules which are available in the market, but for different diseases, whether they can act upon this coronavirus infection or not. So here we, in computationally, what one can do, one has to first select the protein target, that on which target, against which target of the virus you are going to screen out the drug molecules. Here we have taken the main protease, which is encoded by the non-structural protein NSP5 region. I will show you again here. So this is the NSP5 region here, this main protease. Okay, and this main protease, it helps in the polyprotein processing. That means during the process, this region is broken into 16 non-structural proteins. Unless it is not broken into 16 non-structural proteins, the replication will not take place, or the virus will not replicate to produce new viruses. So therefore, NSP5 region, that is the main protease, plays a big role in the viral infection. Hence, we take the main protease impro as the target. This is one of the regions. The other region is that during that period only the crystal structure of the main protease was published. Therefore, it helped us to quickly screen out against this main protease, because it is easier to model if the PDF structure of the protein target is available. Now, as I told you, it helps in virus replication, and it is required in order to prevent the virus infection. It is required to inhibit this protein target. Okay, so among the existing FDA approved drugs in the market, we considered three categories of drug for the screening. One category is protease inhibitors, other category is antiviral drugs, the third category is anti-malarial drugs. So it is quite obvious that we have to consider the protease inhibitor, because we are screening against the main protease. Okay, so therefore the drug molecules which fall in the category of protease inhibitor, that is quite natural to consider them. Antiviral drug, again, since this is coronavirus, so it is quite natural to consider the antiviral medicines available in the market. The third one, anti-malarial, is prompted by some positive results of chloroquine and hydroxychloroquine. So at the beginning of the infection, some reports mention that chloroquine and hydroxychloroquine can cure, or can help to cure the coronavirus infection to some extent. Although today we know that they are not that effective, but the reports that time, it prompted us to consider anti-malarial drugs for the screening. Now here so this is the main protease structure. In the main protease, there is a diet in the structure. You can see here it has zoomed. It is zoomed in here. You can see there are two residues, histidine 41 and cysteine 145. They form the catalytic diet. Basically, they help in the virus replication process by helping in the polyprotein processing. Now, what I will show you some initial screening results. Here we have listed out top 53 drug molecules from the list of 200 FDA approved drugs. Here the 53 drug molecules having interaction energy lower than -7 kilocalorie per mole with the main protease. Some of the So we can see here Bhilpetasvir has topped the list, followed by Glaciaprevir and then Grazoprevir. There are several other drug molecules present in the list, such as Denoprevir, Nilfinavir, Indinavir, uh, Liponavir. So basically, our screening process has faced some important molecules which have been reported to have antiviral effect against the coronavirus, and these are like okay, in the experimental studies, these molecules like indinavir, nelfinavir, denoprevir were reported to be effective against the virus. So this actually tells us that our screening process is quite accurate enough. Here, I want to spend some time on Bhilpetasvir and Glaciaprevir, although not much. Here, these are the two molecules. You can see here Bhilpetasvir and Glaciaprevir, and their binding energies are almost similar to each other. So structurally, you can see Bhilpetasvir. It looks like a long chain. On the other hand, Glaciaprevir, it is more like a circular in shape. Okay, and there are so many electronegative atoms present in the structure, like oxygen, nitrogen, fluorine. Okay. Uh, these are the electronegative elements. Of course, they are also present in the Velvet survey, but it is not much compared to Glaciaprevir. Now, these two drugs are found to have antiviral activity against hepatitis C virus. Glaciaprevir is also a protease inhibitor in some cases. So. These basically, by looking at the structure, and later we have analyzed also the interaction of these two drugs with the main protease, and we have found out there are several interacting residues. The presence of more interacting residues clarify that these molecules can be effective against the main protease, or can be these drug molecules can work on the coronavirus. Although more studies and more computer experimental studies, clinical trials are needed in this direction. So this work we have not published yet, but it has been published as a preprint in KM Archive, a portal. So therefore, since we have not published this work, so I am not going to discuss in more detail about this work. Some studies are pending, so I am not presenting here. Now I will come to target identification for some clinically effective drugs like ivermectin and famotidine. Coming to the first one, ivermectin. Ivermectin is an FDA-approved drug. So it is used to treat various parasite infestations. Okay, it is used to treat head lice, scabies, blindness. Okay, there are several other diseases. One can take ivermectin orally in case of external infestations. One can use this medicine in the skin. Right? So apart from the anti-parasitic nature of the ivermectin, it has been found out that ivermectin is also effective against a broad range of viruses, which include HIV, dengue, influenza, and Zika virus. Now, in the context of coronavirus, the first study was published, which was carried out in Monash University, Australia. A group of scientists, they found out that ivermectin medicine, when applied to the cell line of coronavirus, so in 48 hours, it reduces the cell line by 5,000 folds. So that actually drew the attention of the scientist, demonstrating in that ivermectin could be very effective against the SARS-CoV-2 virus. So following this study, a group of doctors in Bangladesh, they applied ivermectin with another medicine, doxycycline, to COVID-19 patients, and they report some astounding positive results in curing patients. So these reports basically lured us to consider ivermectin to demonstrate its mechanism of binding, or actually on which part of the virus it is going to attack. If we can know, then we can consider that target, and based on that consideration, we can design more more effective molecules which can more strongly interact with that part of the virus, because ivermectin has ensured the inhibition of virus. Therefore, if we can design better molecules better than ivermectin, then we can have better therapeutic. So this is this is our intention, that first we have to reveal that where actually the ivermectin is going to bind or going to attack the virus. Here we have considered several protein targets of the virus. We have considered 12 most important protein targets of the virus, and then what happened? We have taken ivermectin and we have analyzed the interaction of ivermectin with each of the protein targets to see that with which target of the COVID of SARS-CoV-2 it is going to have a stronger interaction. So again, we have performed a docking experiment. We have analyzed the binding energy. The binding energies coming from docking reported here. We can see that among several targets, 12 important targets, and there is another target, important alpha beta 1, which basically helps to transport a virus from one region to the other region of the cell. Against these 13 targets, we have studied ivermectin, and even from the energy values, you can see the lowest energy binding energy is obtained with the RNA dependent RNA polymerase with RNA. In short, it is RDRP RNA. So this lowest energy, it means the ivermectin has the highest, strongest interaction with this protein target, and therefore the initial docking study, it implies that RDRP RNA can be a potential target, but we need to further verify it through the molecular dynamics simulation by stability analysis. So following RDRP RNA, we can see the next binding energies coming from important alpha beta 1, and then the other one is we can see, okay, let me see, and a helicase NCB site. So and this one, so helicase NCB site, it has binding energy minus 9.6. This is the second molecule, second target, potential target, and the third potential target is the important alpha beta 1. So the top three targets we took into consideration for MD analysis. We have analyzed their interaction, and you can see from the picture. So this is the picture of ivermectin interacting with RDRP RNA. This is NCB helicase, and this is important alpha beta 1. So you can see here there are lots of interacting residues, so which basically imply that the interaction of ivermectin with RDRP RNA is quite stronger than other two protein targets. And this is further achieved with the stability analysis in the molecular dynamic simulation. we can see a stable RMSD plot for the RDRP ivermectin complex. So this black color is for helicase helicase NCB site target in complexation with ivermectin. This is RDRP RNA in complexation with ivermectin, and the blue color is for importin alpha beta 1 with ivermectin. So here the stable RMSD plot indicates that the stability of the complex. More fluctuations means the complex is not stable enough. Okay, so similarly, in the root mean square fluctuation of the residues, you can see there is a less amount of fluctuation for the RDRP. Again, okay, this reflects that the RDRP complex is more stable compared to the other top two complexes. Whenever we project the volume of the complexes, three complexes, we got that RDRP complex occupies a less amount of volume subspace compared to the other complexes. So basically, the occupied volume, if it's more, then that means the interaction is not that great or stability is not that great. So smaller the volume, that means greater is the interaction, or greater is the stability, which is okay, which we can see for the ivermectin and uh, DRP RNA complex. Again, from the free energy, we have done also free energy calculation, and we can see for the RDRP complex, we have the lowest free energy, which is of minus 187 kilojoule per mole, followed by importing alpha beta 1, 150 minus 156, and then we have the third one, helicase NCB site, which is 90 minus 94.62 kilojoule per mole. So the other columns in this plot in different colors, they represent the different contributions to the overall binding free energy. The overall binding or total binding free energy is represented by the green color. The other columns of different colors, they are component of the is overall binding free energy. Anyway, so binding free energy analysis and stability analysis further demonstrate that RDRP is the most potential target for ivermectin. If ivermectin is inhibiting or it's working against the virus, it most likely attacking the RDRP RNA region of the virus and thereby destroying the virus. Uh, Again, so this is kind of a conclusive picture of this analysis. So we told that okay, RDRP is the most potential target for ivermectin. Now I will discuss about the next drug molecule, which is famotidine. So famotidine, everybody knows this. This is a gas tablet used for heartburn and several other gastro problems. It's a very easily available medicine in our day-to-day life we use. Talking about the price cost, ivermectin is also a very it is also very easily available at a cheaper price. Now, the study which basically drew the attention first, it is the study carried out in the USA hospitalized patient. A group of scientists, they kept, or doctors, they kept in observation of 1,620 hospitalized patients, and to some of them they offered famotidine. And what found out that famotidine was associated with improved clinical outcomes and reduced risks of intubation or death in the hospitalized patients. So this is the study which motivated us. Then following this study, there are other reports also where basically people have reported that famotidine is to some extent working against the virus. Again, there is nothing known that where the famotidine is going to work, in which portion of the virus it is going to attack. Again, we think that if we can reveal the interaction mechanism or inhibition behavior of famotidine with the virus, it will help design more effective drug molecules in the future. Again, we consider this famotidine molecule and 12 most important virus protein targets. This is basically I have presented earlier. This is the overall viral genome, and then the protein targets, non-structural protein targets are coming from open reading frame region 1A and 1B region. So basically, like earlier, we exposed the medicine uh, against 12 most important virus protein targets. Uh, so we have performed the binding energy analysis, and what we found out that the lowest binding energy or the strongest interaction is found to be with the peptide-like protease of the virus. Um, okay, so if we compare to ivermectin case, if you can recall that the binding energy, initial docking binding energy, it was around minus 10.4 calorie per mole. Compared to that, we have minus 70.9 kilocalorie per mole. So that means, okay, the interaction of famotidine with the protein target. Um, Specified protein target or the most potential protein target of the virus is not that great, but of course among the different protein targets, the peptide-like protease is supposed to be the most potential target for the famotidine molecule. Now we okay, so the prediction has been judged by further molecular dynamics simulation and stability analysis. These are the plots of some dynamical parameters like RMSD, um, uh, radius of gyration, RMA shape. Okay, also we have monitored the number of hydrogen bonds compared to the native protein, that means peptide-like protease. And I want to mention here that the peptide-like protease also helps in virus replication by helping in polyprotein processing, like the main protease. And now, whenever we check the stability of the complex drug protein complex with the native protein structure, we can see that the protein complex, the complex has the lower RMSD value or lower RHG value, okay, lower RMS value. So it these dynamical parameters indicate that after complexation, the protein molecule PL2, peptide-like protease, getting stabilized with the famotidine drug. It assures that whatever the prediction we have made from the docking, that PL2 is the most likely protein target of famotidine. It is supported by the dynamical analysis. And again, you can see that the volume of the protein drug complex is lower than the volume of the only protein complex. This again confirms that the protein is gaining stability after interacting with the famotidine molecule. And there are many hydrogen bonds present in the famotidine and peptide-like protease complex molecule. So these analysis basically support that famotidine, if famotidine has to inhibit the virus, then it has to inhibit the peptide-like protease target. So these two works we have published. I think these two works we have published in book. So this work on the ivermectin we have published in Journal of Biomolecular Structure Dynamics, and this famotidine work also we have published in the same journal. Now we are at a stage to summarize or to make some prediction. So we have revealed that which are the potential protein targets of the clinically effective drugs, ivermectin and famotidine. Okay, from our studies and from the literature report, uh, we have made a proposal, so which has been published in the SES Pharmacological Translational Science, on the title that ivermectin, famotidine, and doxycycline is suggested combinatorial therapeutic for the treatment of COVID-19. So why basically we have proposed ivermectin, famotidine, along with another drug, doxycycline, to be an effective combinatorial therapeutic against the virus. So the reasons of such choice has been briefly or has been touched upon earlier. Basically, we have mentioned that ivermectin. We received reports ivermectin is working on the patient from different countries. Like it first report came from Australia, Monash University, followed by Bangladesh, and a study, a clinical study, was performed in the USA. So in every case, okay, ivermectin basically came with positive reports in curing COVID-19 patients. So therefore, it is quite obvious to consider ivermectin and in the combination therapy. Again, since we have revealed that where ivermectin is going to inhibit the virus, we found out that RDRP RNA, and that is the genetic material which is very crucial for virus replication and infection. Basically, ivermectin is directly going to attack the virus genetic material RNA, and therefore it can basically destroy the whole virus. Now, in the case of famotidine, what we saw that famotidine also acquired some positive reports from different countries, like from the USA, and we knew that famotidine, it is histamine 2 receptor. Basically, it takes care of the acid secretion in the stomach. Earlier, it has been found out that it has some HIV inhibitor. Now, from our studies, we of course saw that famotidine is not going to have that much of binding interaction as we saw in the case of ivermectin. But the positive reports basically then, whatever the positive effects we are getting by applying famotidine to the COVID-19 patient, this may arise either of the two reasons. One is it may inhibit the papain-like protease, which we have demonstrated in our computational studies, or since it takes care of the acid secretion, the healing of the COVID-19 patient and controlling the acid secretion, there might have some link. So either of the two way, famotidine is helping to cure the patient. But still it has to be verified. But since famotidine is working, and we found out that it is targeting one of the important protein targets of the virus, so therefore it is rational to consider famotidine in the combination therapy. The other drug molecule which we considered, this is doxycycline. Doxycycline has well-known antiviral, anti-inflammatory, and antimicrobial properties. So it is a very good anti-inflammatory agent. And we have seen we have received reports on the COVID-19 patients that many patients have suffered from inflammation, pains, etc. Basically, what happens when the infection starts, then cytokines, the inflammatory agents, are produced in large amount, and therefore the patient feels patients feel inflammation due to the production of large amount of cytokines, and therefore it can deteriorate the COVID-19 treatment. Therefore, it is quite justified to include doxycycline in the combination therapy, because doxycycline will take care of all the inflammation or pains or aches, whatever the patient is feeling, thereby helping the other two drugs to boost up their activity. The proposal is this: famotidine will act as a first level barrier, either by reducing the stomach acid or by inhibiting the peptide like protease, which helps in a polyprotein processing. Uh, so as, yeah so This is the case. So whenever the virus gets inside the body, so either of the two ways famotidine can try to put the first level barrier against the virus. If the virus becomes successful to go inside the cell, and the genetic material is released inside the cell, and the infection progresses inside the cell, then if we apply ivermectin, we found out that ivermectin directly attacks RNA material. So therefore, the ivermectin inside the cell will attack RNA and completely destroy the virus. So by applying these two medicines, we can be able to control the virus infection entirely. Doxycycline could be a good augmenting agent. So this is our suggestion. So I have reached almost at the end of the talk. I will conclude that SARS-CoV-2 is very infectious. We designed biomimetic peptides which can inhibit the spike protein of the virus, which can be a potential drug target to treat COVID-19 infection. The repurposing of existing drugs reveals velpetazepine and clesopepimide can be effective against the virus. We found out two important virus protein targets for the two clinically effective medicines, ivermectin and famotidine. RDRP RNA protein target for ivermectin, and peptide-like protease for famotidine. From our computational studies and from the literature knowledge, understanding how the virus infects us, we proposed a combination therapy comprising ivermectin, famotidine, and doxycycline, which can be an effective therapeutic against SARS-CoV-2, which can be one can take it forward for clinical trials officially. So I will end my talk here. So before that, I will acknowledge generously my students who, in the very critical time, in the moment of crisis, they managed to work on such critical biological problem. I want to name my postdoc student, Dr. Partha Sarathi Sengupta, Saroj Kumar Panda, my PhD student, Abhi Kumar, a project student, and Sataranjan Vishwal, project fellow, for their complete devotion to carry out this work, which can be in the betterment of the society. So thank you all. I will stop here, and I will I will ask if you have any questions. If I can show you something, that will be great. So before ending, I want to show you two interesting videos about the virus structure and how the virus basically infects humans. So can I play this video, Professor Ravi Kumar?
Yes, sir. Yes, sir. You can continue.
Yeah.
The World Health Organization was informed of an outbreak of pneumonia of unknown cause detected in Wuhan, China, on the 31st of December 2019. Today we know that the infectious agent was a virus of the coronavirus family, which was named SARS-CoV-2, that causes a respiratory disease termed COVID-19. This virus has not only received widespread media coverage, but also caught the attention of scientists who have been busy analyzing the virus, the disease it causes, and how it spreads. There is already an enormous amount of information available about the virus, and that is why we thought to summarize some of the information in this video. First of all, what do we know about the virus? Coronaviruses receive their name from their appearance in electron micrographs, where the pronounced spike-like protrusions create the effect of a corona, Latin for crown. The genetic material inside the coronavirus is RNA, and the entire genome is almost 30,000 bases long, packed together with a nucleoprotein into the viral particle or virion. For the virus to replicate, however, it needs to insert its RNA into cells and hijack the cellular machinery to produce new viruses. On its surface, the virion contains three main proteins: the envelope, the hemagglutinin esterase, and the spike protein. These are embedded into a lipid bilayer. The large spike protein is responsible for the crown-like protrusions, but it also recognizes the corresponding receptors on human cells called ACE2. The protease ACE2 mediates the cleavage of the spike protein, which then releases an epitope enabling the subsequent fusion of the virus with the human cell.
Okay, I have one more video.
The virus can be carried for up to 27 feet and land on the respiratory system, according to a study in the Journal of American Medical Association. A gene sequence study published in Frontiers in Medicine showed that the coronavirus targets human cells with ACE2 receptors. This means the epithelial cells in the lungs and other organs are vulnerable to the coronavirus. The coronavirus has mutations in the spike proteins on its surface. The mutations to the receptor binding domains, or RBDs, allow the virus to use human ACE2 receptors as entry points for getting into the cell. According to the British Society of Immunology, the virus hijacks the cell and commandeers the structures inside the cell to make more copies of itself. The infected cell's ribosomes are directed to create viral RNA and proteins. After that, the cell's Golgi apparatus puts the proteins and RNA together and coats them in protein, which creates new copies of the coronavirus. The biochemical distress of being hijacked by the virus eventually kills the cell, which causes damage to the infected tissue. At the same time, the body tries to fight off the infection by sending killer T cells. The killer cells search and destroy compromised cells. The immune system attack on the infected part of the body leads to more tissue damage and inflammation. Speaking to The Guardian, John Wilson, president-elect of the Royal Australasian College.
Okay, I will stop here. Thank you very much for your kind attention. I will.
Sir, we have some questions on chat box, sir. Please.
Chat box. So I received. Let me see.
Is it visible, sir?
Okay, one question, sir. How satisfied you are with the two COVID vaccines from Indian side? Okay, so we can see because of the recent appearance of new strains of coronavirus and the reports of further infection from different places all across the world. So this actually signifies that the vaccines developed, they are not able to control the virus entirely. because the virus has gathered some, the mutated or the new strains has gathered some immune evasive nature. Therefore, means for the new strains, still we are not totally safe. Safe even if we use the available COVID-19 vaccines, but they are working fine against the old strains. Okay, but for the new strains, still we are we have to be careful. If I am so, if it's okay with this answer, can I proceed to the next question?
Yes, sir, you can. Sir, another question on the screen, sir.
Which part of RNA produces spike protein? Okay, so it is the spike protein. Spike protein is a structural protein. It is present on the outside surface of the virus. Whether the RNA actually is present inside the virus means inside the lipid bilayer. So structurally, they have different locations, but say whenever the reproduction taking place, okay, whenever replication takes place, then messenger RNA, it acquires certain structural components to make up the full virus. Which full virus, it means that it has a spike protein or all other structural proteins um, to generate a new virus, and new viruses ultimately get released from the infected cell. So RNA actually not directly, you can say that in this portion, but structurally, RNA and spike proteins, they have different locations in the virus structure. uh, Whenever replication takes place, the genetic material acquires necessary structural components. Among one of them is spike protein to make the full replicas of the coronavirus.
Sir, is there any natural products or herbal drugs we
Or
can use for COVID-19 infections?
sure. So there are many Ayurvedic products. Okay, people, when the studies have reported. In fact, one of our studies, which we have carried out, which we have carried out, but we have to yet analyze our results. But there are some Ayurvedic or natural compounds which can be a potential curing agent against the coronavirus. So on the top of my head, I'm not able to tell you that what are the particular compounds, but yes, okay, natural products or Ayurvedic compounds, they have shown, some compounds they have shown the antiviral activity against the coronavirus in the recent studies.
Okay, thank you, sir, for your nice presentation. You have clearly mentioned everything related to COVID-19 and its treatments, and also focused on drug repurposing, which is most recent trends in the current scenario. We are very much thankful for you, sir.
Thank you very much. Thank you, Vivian.
Thank you so much, sir, for this informative session. Now I request our beloved principal, sir, Dr. V.V. Ravi Kumar, sir, to kindly present certificate of appreciation to sir as a part of gratitude and appreciation from Roland Institute of Pharmaceutical Sciences, Barampur.
Thank you once again, Dr. Rana sir,
Thank
for
you.
accepting our invitation and sharing your knowledge. It was really a wonderful session. I hope all the audience were benefited by the information given by you, which is very much related to the current scenario and also link it to the theme that this FDP is being conducted. Thank you once again, sir. On behalf of Roland Institute of Pharmaceutical Sciences, I request you to kindly accept this certificate of appreciation.
Thank you. Thank you for this certificate of appreciation. I, I acknowledge the acceptance of this. Thank you very much, Professor Ravi Kumar, and the organizing committee members for such for inviting in such nice session, nice program. I look forward to having such kind of interaction in the future. So,
We also want to have you in our college physically also, sir, for some information
yes,
to our
you.
students also, sir.
Whenever the situation will permit, okay, we will certainly visit. Okay, I will look forward to visiting your institute in the future.
Okay, sir. Thank you, sir.
Thank you. Thank you. That's good.
So, second session will be conducted at 4 p.m. Please stay here. So, today's speaker will be Professor Nami sir from Australia. So, he'll join at sharp 4 p.m. Please stay online.
Hi, Navi. How are you?
Hi, Ravi. Hi, Ravi. Can you hear me?
Yeah, yeah.
Okay.
How are you?
I'm fine. I'm fine. How are you?
I'm fine, thank you. Bari formal day only.
We have to do
Procedure.
it.
Yeah.
Is my
Up
audio
to.
okay, or do
Yeah, yeah.
is my audio
It's
okay?
okay. It's okay.
It's okay. Right.
Mm. What's the time now?
It's 9:25
Okay.
p.m.
Yeah. Means you're always ahead, na? In studies or timings.
So, what
Shall we start?
is this the last session for you, or
Yeah, yeah.
so 4 to 5?
Last one.
Okay. And is today the last day of the program, or?
No, no, no. Tomorrow we have one more
Uh-huh.
session.
Okay.
So how is everything?
Oh, everything is fine. So busy days. I, in fact, didn't get any time to prepare my slides. I asked my student to prepare them, and I received them this evening.
Hmm. This is how means teaching pedagogy
Yes,
works also.
because
Na?
our semester starts from next week, and we
Okay.
have
Sorry for
thousands of
troubling you.
students.
Uh, you told na you're over busy.
Yes, so
Completing your
usually
assignments.
just a couple of weeks before the start of the semester and a couple of weeks towards the end of the semester, that's a peak period for us.
Okay.
And we don't have any seats limit in our courses. So we have like up to 1,000 students in each class.
Okay. Online. Online
Uh,
course.
on At the moment, we are doing online, but regardless of online or offline, we
Hmm.
always have more than 500 students in each subject.
Okay. Hmm. Let us start then. Huh?
Okay, yeah. So, how should I
No, no.
actually share my screen? Is there any way to do that?
It is there. Na? You just see at
Okay,
the
yes,
bottom of
yes.
the
Right,
share
right.
is there.
Because, yeah,
No.
usually I do it from Zoom.
Okay.
Um,
Sir, you come on. Okay, madam, you can start.
Okay, sir, fine. Good afternoon, everyone here. Welcome to the second session of day 9. Today we have with us Dr. Professor Srinivas Nami, sir. Prior to his talk, I would take this opportunity to introduce sir to all of us. Presently, sir is working as a senior lecturer in pharmacology and academic course advisor, medical sciences at Discipline of Medical Sciences, School of Science, Western Sydney University, Australia. Dr. Nami holds a doctorate in pharmaceutical science, specializing in pharmacology from Andhra University, India. Before joining the Western University, Sydney, he joined as a faculty of pharmacy, University of Sydney, after gaining postdoctoral experience as that fellow at the University of Tübingen, Germany, and at the University of Manitoba, Canada. Dr. Nami also worked as head of pharmacology in an industry where he was instrumental in establishing a new pharmacological research laboratory. Dr. Nami has distinguished track record in Ayurvedic and Western herbal medicine research for more than 20 years and has made significant contribution to both basic and applied research related to drug discovery, biological evaluation, and mechanistic studies with a special focus on metabolic disorders. Dr. Nami has attracted competitive research funds from national ARC, AISRF, and NICM, and also international like Germany, that fellowship. He has one international patent and published over 90 scientific papers, 10 editorials, and 3 book chapters. Dr. Nami's research has been cited so far in over 3,000 publications with an H index of 30. He has supervised 8 PhDs, 8 masters, and 15 honors projects. He is serving as secretary for the Australian DAD Alumni Association. He is frequently invited to talk and chair at national and international conferences and to educate PhD thesis from Australia and overseas universities. He He is an international expert reviewer of a number of national and international research grant bodies. He has been awarded a number of fellowship and awards, including the prestigious DAD Fellowship Germany and SESQUI Postdoctoral Fellowship Australia and CSIR Senior Research Fellowship India. He has been honored with Marquis biographical listing in Who's Who in Science and Engineering, Who's Who of Professionals, Who's Who in Asia, and Who's Who in the World. In the year 2018, he has been awarded Research Excellence Award by the School of Science and Health, Western Sydney University, for his outstanding contribution in research. With this, I now request Sir to kindly continue with the session.
Thank you, Gina, for your kind words.
Sir, please make full screen.
Can you see full screen now?
Just
I can see
check.
full screen on my window.
Just sideways it is in PowerPoint mode, sir. This third option you can choose. One chap is Basun. Sir.
How is it now? Could you please check? How is it? Is it fine?
It is fine, sir, but you click F5, sir, otherwise.
F5.
This slide PowerPoint mode is there after this percentage, before percent. It is okay, but it is visible, sir, no problem.
What do you mean by PowerPoint mode? Like, are you unable to see full screen or?
Here you have 92%, sir. Before that one option is there. You can click that.
Where is that one? Okay. Let me try.
Yes,
You see the bottom right. Right. Now the screen has gone.
Is it fine now?
You click F5 in your laptop. F5.
F5, yeah. I did it.
Take this, sir. Sir can continue like this. No issues.
You see the bottom right of your screen. That some percentage is written, 100%.
Yeah, at the moment it is 100%.
You just come to the icon left to 100%.
200.
No. Icon. 4 icons are there, na?
Oh yeah.
You press the icon immediate left to 100%.
Yeah, I did that.
Okay then. Okay, it's fine. It is visible.
Yes. I went to full screen mode on my computer.
Okay.
So on my computer, it's all full screen.
It is okay. It's coming.
It's okay? Okay, shall I continue then?
Yeah, please.
Okay. All right. Good evening, everyone. As Dr. Gina introduced me, my name is Srinivas Nami from Western Sydney University. I was, in fact, originated from India, from Andhra University. I did all my studies in Andhra University. And I would like to um, extend my special thanks to Professor Ravi Kumar, who has kindly invited me to provide this lecture for your faculty development program. And despite my busy schedule, I eventually made it. So thanks for inviting me, Ravi Kumar. And without wasting our time, I would like to get into the details of today's talk. As I said, we are in a challenging environment at the moment. We have seen for the last 1 year or so, we have seen so many devastations due to COVID-19. It has caused troubles to each and every one across the globe, to every individual, to every industry, including the education sector. We have seen how each of those educational institutes, right from primary schooling to tertiary educations, they have suffered a lot. And eventually, it was a challenging time, not only for the students, but also to every staff member in educational institutions. And we have seen that from the UNESCO statistics, more than 1.5 billion students have been engaged in remote learning over the last 1 year or so across the globe. And students have faced a lot of challenges in doing that remote learning or online learning, especially in the underdeveloped countries and developing countries where students were unable to access internet and technologies. So they faced a lot of troubles, and students in those um, remote areas have used their mobile phones and smartphones and other mobile devices as online platform in order to continue their learning. However, some students, they're fortunate enough to have laptops or desktops or other digital devices for their learning. But we have seen lots of challenges during the last 1 year due to COVID-19 pandemic. And not only students, like I said, faculty members, they took up the real challenge ever since we have seen the pandemic in 2020, beginning of March, it seems. So since then, they have taken some acute measures in order to continue the delivery of teaching and then bringing um, the students' life back to normal. But what we have seen um, as of now is we don't have the normalcy again. Like we are thinking that new normality, but we are not going to have the back to normal situations until some point or towards the end of 2021. What is that? I've written some errors there. So it's until the end of 2021, we may not see any normalcy in the education sector. But we have seen challenges not only in education sector, in every industry. Governments have faced a lot of troubles. They have extended their support to every citizen in every country. Um, and they have given some relaxations to every industry as well. So what we have done in terms of faculty, when we took up those acute measures at the beginning of COVID-19 is majority of the faculty members, they were not prepared for those challenges. They didn't knew that COVID-19 is something that is going to affect their lives. So they haven't prepared for that. So they haven't prepared for any online mode of teaching. So it was entirely a new challenge for them in reality. And they tried to explore what online teaching is really meant for them and how that new pedagogy of online teaching really looks like when it comes to the online mode of teaching, and how that actually engage the students in the remote learning, and how best that online learning can actually produce the results what they expect. So they have done to the maximum extent what they can do, but in that process most of the faculty members, what they have done at the beginning of COVID-19 is they tried to replicate the normal classroom environment to online environment. Even in advanced countries, most of the faculty members did the same thing at the initial stages of COVID-19. And everyone knows that those immediate changes were not practically viable because some of the learning environments were not efficient for the students. Students were not engaged in those classes. And in fact, we have seen lots of cases regarding that, and we have seen some feedback as well from the students. And also, students, not all students can actually access synchronous classes. As I said before, students may not be having the internet technologies, and there may be many factors. People cannot afford internet, broadband internet. They may not be having digital devices to have the synchronous teaching and learning activities. So they have faced some troubles in that process. And faculty members also began to discover what has been known for some time in the teaching and learning pedagogy in the literature, and also some of the practices that have been used in many institutions over the time. So teaching and pedagogy is not entirely new to COVID-19 pandemic, but we know that there were variety of teaching methods and practices adopted elsewhere. And the instructors also began to experiment with their own personal challenges and try to implement small group work practices or project-based learning, sometimes recording of short videos in order to deliver them via the learning management platforms. And sometimes faculty members also sought help from their colleagues who have known to use internet technologies and other digital technologies, and who have got some proven records to show what is working and what is not working within their disciplines. So that was the acute measures taken by everyone across the globe in every educational sector. But we know that we are not coming back to normalcy very soon. So we need to tackle all these challenges for the rest, at least for 1 year. And also post-COVID, we may be having some digital technologies embedded into the traditional mode of teaching. There will be some new pedagogies that are coming into place depending on the policies of each educational institution. And there may be many changes which are actually triggering that online learning environment as a result of COVID-19, and some technologies, new technologies, which increase the access and flexibility of teaching and learning for the students, and also some technology-led pedagogy changes that we are going to show you in a while. Many institutions across the globe, they started exploring these developments even beyond the pandemic. In that way, they are trying to use or maximize their way of online usage in their teaching and learning environment. There will be some implications for these, for the students as well as for the faculties and institutions, which highlight some developments in online learning and also the impact of these technologies in our understanding of teaching and learning due to the new pedagogy. So some of the key developments that trigger the development of new pedagogies are actually based on demands of knowledge-based society and also the expectations of the students in the new modern era, and also the availability of various technologies, and lastly but not least, the fast-changing world in the work sector. So we are going to look at each of these, how they actually trigger for the development of new pedagogy. So looking at the knowledge-based society, for example, We know that in the traditional way of teaching, the instructors or the faculty members used to provide big blocks of lectures for 1 hour or 2 hours and then give all the material to the students. And students are forced to learn all those materials within a short span of time in order to reproduce that in the assessments. But in the knowledge-based society, what we try to do is we don't have to give all that material in a block. Rather, what we can do is we can also help students to manage their own knowledge. And in order to gain that knowledge, we can give materials where they can actually find the solutions, they can analyze the information, evaluate it, and then apply and then practice that knowledge. And we have also seen that when you extend that into the research environment, for example, knowledge is not something that is static, it is more dynamic in nature. So we have seen lots of developments happening. So knowledge is continuously expanding. And on average, we can say that 1.8 million papers are published annually in over 28,000 scientific journals. And India is not very far. India is really doing very well. So annually, India is growing at a nearly at 11% in terms of research, which is good. So that continuous development of knowledge and how we impart that knowledge into the students' mind, that's important. And the second thing is like in the 21st century society, how we can apply that knowledge. So through various activities which can promote soft skills such as critical thinking, independent learning, the usage of technologies such as the information technology and software, data analysis within a particular discipline, and also, but not least, the entrepreneurship in order to expand their knowledge and then do something to the society. And like I said in the first point, the continuous development of new knowledge. So we need to educate the students for lifelong learning rather than giving a big chunk of material for that semester or for that year. So we need to provide information to the students so that they can actually get those graduate attributes or soft skills in order to manage their own learning throughout the life, and also to produce skilled workers for the workforce. So in terms of the student expectations, in the 21st century, for example, we know that technology is actually driving. Students think that technology is close to them, it's part of the environment. So they expect a lot in terms of what they can do with the technology. So they think technology can actually teach them. But the instructors or the faculty members in institutions, they are the driving force, and to show the path to the students how to get that excellence in teaching and learning. But what we see from the students' expectations point of view is there is a lot of change in student demographics in educational sector. As we see school leavers, even though they are a big group in educational sector, slowly what we are going to see is that more matured students coming into the coming into the educational institutions for further learning, or sometimes they want to combine their work along with studies. Sometimes they want to upgrade their skills. So they need a kind of flexible learning environment to have those flexible learning options. And we have seen that in India, due to the latest national curriculum policy that was passed in the parliament, in future we may also see matured students coming to Indian institutions rather than direct school leavers coming to the educational institutions. So we need to be prepared for that change in demographics and for the matured mindset of the students, and also to provide a pathway for their work as well along with their studies. And We also need to find out what students are actually expecting in terms of their career options. So they always see whether they get good jobs after 4 or 5 years of their studies. So everyone expect a good job after their studies, and most of the times they want to apply what they have learned in their workforce rather than jumping onto different fields. So most students, they want that, they want to apply what they have studied, and um, they want to see that they they have their skills upgraded continuously to be in the job force for sustainable period. The technology, that's what the students always love for. So they want to see the technology usage in teaching and learning in order to develop essential information. They want to use the technology. Sometimes they use various digital technology platforms in order to get the information what they want, and they want to become fluency in the usage of technology as well. So when it comes to the actual technologies that are available for students to use, we have seen in the modern era, social media is one of the platforms where students meet with other students and also share information in a more um, liable way. And various mobile devices, they use their smartphones or um, laptops or other notepads, etc., in order to share information. And what is important with technology is that it increases the access to the information, and also using the devices they can create information, and also they can edit, they can share that knowledge with other fellow students and with faculty members, and that in fact increases their motivation and engagement sometimes with the studies, as long as they are using technology in the right way. Added to those new technologies, we also saw artificial intelligence and the involvement of artificial intelligence in teaching and learning environment. So we are going to see a lot in the next few years on the usage of artificial intelligence in various teaching and learning pedagogies, including assessments, automatic assessments, etc. And we also knew that traditionally we have been using various simulations in various disciplines, animations, games, and other online resources, etc. So all these technologies can be used in order to develop a new pedagogy, not only during the COVID, but also after COVID. One more point that triggers for the necessity of new pedagogy is the changing world. So the workforce is changing across the globe, and the policies are changing across the world. So that leads to lots of changes in the nature of work, how it is done, and how it is actually organized. So there is a lot of change towards project-based work in many sectors, in many industries, and we have seen the flattening of organizational structures. So traditionally, we have seen a vertical hierarchy of the organizational structures, but nowadays it's more flattened. So the projects are actually distributed more horizontally. So we have more projects in a widespread group, and lot of relationships between the human and the technology. So in fact we have seen decline in personal relationships, we have seen increase in the human to technology relationships in the modern era. So that's going to continue, unfortunately, but that's part of the changing world. And also due to changes in the global networks and the supply chains, so we need to have that sustainable workforce, and also to meet the competition within the supply chains and global networks. We need to adapt to those situations. And other point what students consider is anytime and anywhere learning. So students they look for more flexibility nowadays. So they want to see they can learn from any remote area, from anywhere where they live, and at any time. So it's not like always a synchronous mode of delivery where students may or may not be accessing that at that right point. So they look for asynchronous way of delivery, such as putting that information on a learning platform or recorded videos, etc., so that they can actually go through the materials at any time as per their convenience. And lastly, the upskilling and reskilling of the um, graduate attributes. So students and graduates need to upskill their knowledge and also reskill if they want to change to a new job environment. They need to get those new skills. And what the universities and other educational institutions are doing nowadays is to have those short modules which can actually provide those upskills and reskills, and also provide an opportunity for the students to get some micro credentials for those modules when they start continuing in future. For example, in a full course, they can add these micro modules into their full length modules to get some micro credentials for the previous modules of study. So those things are also happening, and there were some universities are actually giving those micro modules and micro credentials. So continuing with those contributing or triggering factors, we can see the various elements that actually help in developing of the new pedagogy in the modern era. What we can see is, you can see there are 7 points here. I'm going to talk about all these 7 points. Blended learning is one of
Sir,
the.
excuse me, sir.
Sorry, yes, can
Sir,
you hear me?
slide is not, sir,
Sorry,
slide is not moving, sir. Please change the
slide.
slide, sir. It is in second number slide.
Is it?
Yes, sir.
I'm,
Please
I'm,
change.
I'm in page, I'm on page 10.
Okay, but it is not moving, I think, sir. Please check.
Okay. It's actually moving on my screen.
Okay, but here it is not showing, sir. That's why.
Are you still on second screen? Second screen is the starting screen, isn't it?
Yes, it is there only, sir.
You see that image, education image and online learning is a key contributing factor. You still see that?
Yes, sir.
I didn't know that. I was actually moving all these slides.
Yes, sir.
Okay, so should I stop and then again start it, or?
Yes, yes, please try, sir.
Okay, so I'm stopping the sharing, and I'm going to share it again.
You go to first option, sir. Share first option you go.
Sorry?
In share screen, you select first option inside.
Okay, okay.
Your entire screen. First option is your entire screen.
So should I select entire screen?
Yes, sir.
Okay. So what you see now?
Yes, sir. Not came, sir. Not came, sir. Again, you select.
So first option is entire screen. Second one is application window.
So first one you select, sir, entire screen.
Okay, if I have selected the first one and then share. Uh, so you see my entire screen now.
No, sir, not came, sir, till now. Just. You make it full screen in your desktop, sir. Not came, sir. Again went back.
Okay, so I have full screen on my computer,
No,
but
sir.
in order to open the StreamYard
It's
window,
in our system. It is not showing, sir, now.
wait. How about now?
Just a second.
Can you see it?
Yes, sir. It is visible now.
Okay, so
You can change your slides.
yeah, so you see in full screen now.
Yes, sir. Yes, sir. You can change it now.
Okay, so the screens are moving, right?
No, still it is stagnant one place.
So it's not moving there.
No, sir.
I can move them here. So what's actually stopping that one?
Slide mode नहीं हो.
How about now?
Sir,
Yes or no?
not coming, sir, now.
Not coming.
No, sir.
Hello, Nami.
Yes, Ravi.
Can you send us the presentation so that we can operate from here, and
Okay.
you send me to the mail ID from which I send the
Okay.
invitation. Can you?
So,
No, other. Sir,
okay, I'm sending the presentation to you. Okay.
In that same mail ID, sir, what I sent you the StreamYard link.
Uh-huh.
where you received the link for
Yes,
today's program.
I'm doing it to the same email.
Okay.
Thank you, sir. We received, sir, your presentation.
Okay. Can you hear me now?
Yes, sir. Yes, sir.
So should I open my presentation on my computer and then continue, or?
No, sir. I'll show you, sir. You check once. Sir, it is visible to you now?
Yes, but it's not full screen for me. It's a small window.
Okay. Just a second.
Yes, now it is full screen.
Okay, okay. I stopped it again. Just a second, sir.
So will you be moving the slides?
Just a second.
Okay.
Now, sir, it is visible to you, sir?
Yes.
So I'll go to 10 number slide, sir. Slide is moving, sir.
Yes, moving.
Yes, yes. So I'll go 10 number slide, sir.
Yeah, please go to slide 10.
Okay, sir.
Yeah.
Continue, sir. Thank you.
Yeah.
Same link, na zero. Madam. Sir, you continue, sir.
Okay. Sorry everyone for that breaking. So like I said, these 7 elements contribute a lot to the development of any new pedagogy. And we see blended learning is one of them, combined with collaborative approaches for constructive knowledge, multimedia, and open educational resources. Student control, choice, and independence is another factor. And also we see that flexible learning through anywhere, anytime, any size learning. The kinds of assessments that we are going to include in the new pedagogy. And last but not least, we also have the self-directed and non-formal learning for a range of students, including the mature age students. So next slide, please. So looking at the blended learning and how it actually contributes to the new pedagogy, we know that in the traditional mode of teaching, we only have a clear distinction between classroom teaching and online teaching. In classroom teaching environment, we know that teachers provide lectures and coupled with some practical classes or tutorial classes and providing some online resources for the students, and then going for the assessments towards the end. Whereas in complete online teaching, we know that everything was delivered online, including the teaching materials, lectures, and sometimes online practicals and online exams as well. So in the blended learning approach, as most of the institutions are actually doing it, it's not a new thing that we are actually inventing or discovered due to COVID. So where the classroom and the online teaching environment are combined. So the classroom time is in fact reduced, but it's not completely eliminated. So we still use some of the classroom environment coupled with online environment. And the faculty and instructors, they need to decide on what are the elements that can be included in the classroom environment and what are the various learning environments that can be used in the online teaching. So within the same blended learning approach, in recent times, we have seen a lot about flipped classrooms, which is nothing but just reversing the traditional way of delivering the content. In the traditional way of delivering content, what we have seen is the lectures are actually given first, and then students take some homework based on the lectures and then do some quizzes or other practical exercises or maybe some tutorial works or workshops. So the lecture comes first, followed by some additional learning activities. In the flipped classroom model, we reverse that teaching approach. So students do some pre-readings. So the pre-readings are actually coming from pre-recorded lectures sometimes. Sometimes there may be some other materials, some online resources are provided for students or animations are provided. Students need to go through them and then come to the class to have some interactive discussions. So during those interactive discussions, students can have a range of activities, including case studies or doing some workshop activities, maybe some peer group discussions on the subject matter, and so on. So the flipped classrooms is one of the new additions into the blended learning approach. And like I said, the faculty or the instructors, they need to be
अभी अंदर जाएंगे तो डिस्टर्ब हो जाएगा सर, अंदर जाएंगे तो डिस्टर्ब हो
so
जाएगा.
the faculty or the instructors, they need to be looking at what will be the successful model for them in terms of what actually best for online activities and what is actually best for on-campus activities. So they need to determine about the division of activities for the on-campus and online environments. Next slide, please.
Yes, sir.
So we have seen from the early days of online teaching, there was some emphasis on enabling students to construct knowledge through various.
बंद करूंगा, आप नेक्स्ट स्लाइड पर कीजिए. अभी चल रहा है, तब बोल देंगे तो करने का.
I'm getting some disturbance.
कुछ डिस्टर्ब नहीं करना.
So there was an emphasis on enabling students to construct the knowledge and also build some communities of inquiry through the various practice approaches, and also through questioning and discussion, sharing of individual perspectives and sources of information between the peers, and analyzing various online resources. And the instructor usually gives the feedback at the end. So such kind of collaborative approaches in order to construct a piece of knowledge rather than giving a big chunk of lecture at the beginning is one of the effective pedagogy's development. Coupled with that, the social media, which has encouraged the development of community practices. So students use social media where they get sharing of experiences from recent graduates and people working in the workforce. They share their experiences on a particular topic of interest, and students discuss the theories and challenges coming out of those real life experiences. And in that way, they can learn from each other. So that's another collaborative approach of learning. And the contributions and reflections from the expert people, from the wider public. Often the instructors and faculty members, they try to include external experts within that discipline into the delivery of the courses. So external experts interact with the students through the learning platforms or through social media, or sometimes they even come to the um, on-site to deliver some lectures and give their insights into that content. So that actually helps not only getting knowledge from the external experts, but also provide important contacts and network building outside of the institutions. So that would help the graduates when they complete their studies. They know whom to get in touch in order to get into the job force. Next slide, please.
वो कहाँ था वो छोड़ आए कुछ टाइम के बाद लैग नहीं मिलता है ये यूट्यूब और ये आएगा ये आप नेक्स्ट किया ना आएगा सर ये आ गया वो कहाँ मम्मी कहाँ नहीं दिखेगा इधर नीचे है आवाज आ रहा है ना हमको देखिए अभी तक इधर दोनों आ रहे हैं ना अभी तक दोनों आ रहे हैं ना आप लॉग इन किए क्या
And sometimes these educational resources can be embedded in the core course content to be able to provide to the students along with the main lectures and other resources. Sometimes they can be used to target the students who are actually struggling to understand the content. So they can be used in different ways. And what is important and what is an advantage with these open educational resources is students can study them free of cost. They can reduce their cost of learning. Coupled with the digital media and open educational resources, we have seen lots of publishers nowadays, they are moving towards the e-text format rather than the traditional handbook format of their textbooks. And we have seen e-text, not only just PDF version, but also e-text version where you can actually see the interactive animations and videos and also audio clips, etc., and some graphics. And students can do some annotations, they can do some additions and changes within the e-text environment. And it is very helpful for the instructors to create assessments because publishers, they do have platforms like test generators. Every publisher nowadays, they have their own test generator and they provide the online resources to the instructors. I don't know how many of the faculty members are actually using those resources from the publishers, but keep in mind that for every prescribed textbook, you are entitled to get all those online resources from the publishers. So if you contact your local publishing company contact person, the area manager or someone, they will be able to provide lot many resources for every textbook that you use. And they have the test generators which can be used to create assessments in an easy way, in a format that is required to be used in any environment, in offline environment or online environment. And also the e-texts are also they also provide an opportunity to provide feedback directly to the students through the e-text learning. So that could be a useful opportunity for the instructors. Next slide, please. So from the student point of view, like I mentioned earlier, student need to have a control on their learning and choice of learning, and they look for more independence nowadays. Internet resources are available to students. They can access to any content um, from multiple sources. So students get that piece of information from multiple sources, so they can actually um, decide on which content they need. And also the mobile devices what they use, the smartphones, and students often do have video cameras where they can collect the examples in digital format and data, and that can be shared between the students and also between the students and the instructors. So that's giving a more flexible approach for um, content selection as well as content choice and delivery as well as the assessment of the content. And the use of internet resources also educate to students to take responsibility of their own learning. So instructors also need to educate students so that students actually take ownership of their learning and responsibility of their own learning. So they need to be more matured about what they are learning from internet and so on. So that leads to changing roles of the instructor. So it's not always the instructor is giving all the information to the students, but in the changing environment of teaching and learning, instructor can actually take a role of a facilitator rather than an instructor, where they can actually facilitate the learning of the student by guiding them to show the required information, where they can get it, and what sort of technologies they can use to get that information and so on, rather than instructor giving all that information to the student, and student is more kept in a passive environment. So student needs to be in a more active environment, and student needs to be in a more proactive way of getting the information. Next slide, please. So students often look for anywhere, anytime, any size learning. What it means is, when you look at that any size learning, it's all about creating small modules and giving small instructional videos to the students so they can learn those small modules that can be implemented into their job force immediately, rather than actually going through the entire content of the unit. And these small modules, once they complete, and then once they complete the assessments, can be used to get micro credentials in order to use them in their later studies if they want. So such kind of small module studies are in place in some universities. And the usage of mobiles, smartphones, tablets, and other digital devices, they are very helpful in having um, learning from anywhere and at any time as convenient to the students. Students get their content um, from internet or from the learning management systems of the institutions. They can get all those resources and quizzes and the discussion boards where students connect to each other to share their knowledge. And the discussion boards are also very helpful, coupled with e-portfolios that I'm going to talk in a while, that can be used in a form of assessment as well, to say how actively students are engaged within that content. Coming to the self-directed and non-formal learning, this is where the matured age students, or students who are not um, directly admitted into a particular course, they get an opportunity for that self-directed learning using various interactive resources and YouTube channels. And such kind of non-formal learning can be used when they get into the official course um, in the later period. And the open educational resources like what I mentioned, like small lectures or animations, some free simulations and virtual labs that can also be used for self-directed learning. So the important aspects of teaching and learning, as we always see, is the challenges in the assessment, and lots of feedback has been gathered during the last 1 year from many institutions in terms of online learning and online assessment. So there are some positives and negatives as well. So there should be some new ways of assessing the knowledge in the new online learning environment. Opportunities like discussion groups for students, they can be provided in the learning platforms like Blackboard or any other learning platform, where the e-portfolios for the students can be created individually and also for the groups, where students interact with their group members on a particular subject matter, and that can be assessed. So that can be actually included in the assessment criteria, and a portion of the mark can be given to that. So based on those active discussions between the students, the assessments can be completed. Peer assessment is one of the ways of engaging students within the content and also getting to know about how to assess the fellow students' work and how to actually learn from that assessment in order to enhance the learning. The usage of learning analytics, there are a number of learning analytics available. Some are free and some are provided free of charge by various publishers. And student mobile phones can be used. So a small app can be downloaded into the mobile phones, and using these analytics, we can constantly have the engagement of the students right from the beginning in the subject rather than assessing their knowledge towards the end. So various ways of assessing that knowledge right from the beginning. In the early classes, instructors can provide some questions within the lectures on the lecture content and post them to students' mobile devices in real life, and get the answers from the students and analyze that to see who is actually following the content and who is actually behind. So that actually helps to push the students who are not able to understand the content matter. And coming to the next point here is the competency based assessment rather than simply the learning material based assessment. So majority of the universities are actually moving towards the competency based assessments nowadays, and also that is something that is pushed by many governments as well. So students are assessed on the competency of a particular skill rather than the theoretical knowledge, what they gain in the content. Sometimes on-demand assessments also help the the assessment of the student engagement on the content, not just at one point, but it can be done at multiple points at any time during the semester. For example, students should be in a position to perform in that assessment. So the on-demand assessments are also one of the new pedagogies nowadays. Coupled with those, there are many in the literature. I'm not listing all of them here, but there are various types of assessments that can be used in the online mode as well. And most of the universities are actually going for online quiz type assessments nowadays. I'm not sure to what extent that is actually reliable, but if you ask me, quiz is just like a platform that can be used for not only for exams, for anything. For a survey, for example, you put the survey in the form of a quiz. So conducting an exam in a quiz format is sometimes not very effective, and also the cheating among the student groups during the quiz exams is very high. And nowadays there are third-party proctoring companies coming onto the market. Those proctoring companies actually conduct the online assessments in collaboration with institutions. So that is one of the effective ways of avoiding contract cheating in assessments. But again, there are other inexpensive ways of doing assessment without any cheating. For example, we use sometimes Zoom, where students need to sit in a closed room, and students need to have their camera open, where instructors can actually see the entire room, and student is allowed to sit in the room without any textbooks, and the student desk has to be visible to the instructor. So so a group of 10 to 20 students for each instructor will be allocated during the exam. So that's one way of doing, if the institutions are not able to afford the third party proctoring assessments. And again, from the um, instructor's point of view, how to actually generate all those assessments within a short span of time. That's another challenge for the instructors. So, like I said, the publishers have got the assessment generating software like Test Generator, and there are some free test generators on internet that can be used as well. But I don't recommend to use all those free ones, because whatever assessment that you prepare using the free test generator, it may be available on the public domain. So the usage of publisher test generator is recommended to generate any kind of assessment, not only a multiple choice exam, but also a short answer type exam, um, or any kind of open-ended question. You name whatever, you can do there. And the marking of student assessments, depending on the size of each subject. Some universities may be having large size units, some hundreds of students in each unit. So the marking is another challenge for the instructors, and the kind of assessment they conduct um, determines the assessment marking time as well. So if it is just a quiz exam, it may not be taking that much time. If it is a short answer, very short answer, that is still fine. But when it comes to essay questions or any other questions, then they need to determine how they're going to mark them. So there are some automated marking of assessments happening through various platforms. So that happens mostly for the quiz type questions, but not for the open-ended questions. Adaptive assessment is something that can be tailored to individual student or individual groups of students, where they can um, um, assess the individual student based on the any special criteria for that particular student, or special considerations that may be allowed to that student, and any other academic integration programs or anything like that, if there are for those students. So all those various elements can be used in the development of new pedagogies of teaching and learning. And combining all those elements, so we come to an emerging 3-point trend here. What you see here is moving to open up learning, which means making it more flexible, like blended learning and flipped classrooms and other online learning environments, and increased sharing of power between the instructor and the student. So like I mentioned, the instructor is not instructor anymore. The instructor can actually drop the role as instructor and become a facilitator or a guide to the student to keep the student active in that program, and get all that information by themselves. And the technology, the usage of technology, and the kind of technology that can be used in teaching and learning. Next slide, please. So that leads to the implications of the various element and technologies and leading to some questions and how the instructors are going to change their teaching and learning pedagogy. So there will be some implications for the faculty members. There will be some impacts on the students as well, and also for the institutions. And it's not new, like I said. We are not doing any innovations in teaching pedagogies just because of COVID. Even before the COVID-19 era, so developments in teaching and learning pedagogies, they were constantly going on, and institutions kept on implementing those pedagogies and then getting feedback from those practices and then keep on moving. And the various innovative applications of technology that are available nowadays, they can be used into teaching and learning, and to see how best they can be used in the development of materials, and then how best they can be used in the evaluation of those materials. The remote teaching, especially during the pandemic, and that was closely observed, and then lots of positives and lots of failures as well, like I said. So 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 we take all those positives, what went well and what didn't work for some. So we take those failures and then we keep on implementing new strategies. And the experience of teaching in synchronous platforms, Zoom or Adobe or Google Meet or Microsoft Teams, etc., and how best students can actually cope with those synchronous platforms, whether students like them or not, whether students are able to um, um, get in touch with those synchronous at the right time or not. If not, what are the various asynchronous platforms like the Blackboard, Canvas, or Moodle, or other learning management systems that can be used, where students can actually get to that material at any time, anywhere. And based on these various questions that can be asked by yourself in adapting all these new pedagogies, and how best that can be implemented to the students. Next slide, please. So in the design of any new pedagogies, we need to find out what are the new factors that we are taking into account in the teaching and in our course design, and what elements of classroom practice actually you maintain, and what you have learned about student needs, and what they actually prefer, and what are the concerns, and what's the success rate with online learning. And for example, in our university, in my own subjects, I don't like online learning. So I need a blended learning approach where some of the learning and teaching activities can be done on campus. And especially, we don't like the online examination. So students, they still cheat in the exams regardless of um, what security measure that you put in place. So we need to look at all those success rates in online learning compared to offline learning, and what actually surprised you and what actually delighted you, and how you're actually leveraging with the emerging research on how students learn, and the importance of students co-creating knowledge through various project-based works, group-based works into that learning design, and what specific strengths and limitations for online delivery are linked to the subject matter that you teach, and what new approaches to assessment you are looking to explore, and adopt any new designs focus on continuous or authentic assessments. So different types of assessment practices are available, continuous or authentic. So you can actually dig into that information to find more information. And in terms of student learning and the impact of these new pedagogies on student learning, we need to see what new demands are students making in terms of how they want to be taught and assessed, and what are your responses for those. What are students saying about the conditions under which they are experiencing online learning? Are there any limits to access the information or limits to use the technology, and the technological skills of the students, for example, which impacts their learning environment? And what new roles are students taking in the online or hybrid mode of learning, the blended learning, and how this has changed your teaching practice? So we need to get answers for all these questions before we start thinking about um, doing any new changes in teaching and learning. And what new areas of student support are being built into course structures to facilitate effective online learning, and what new strategies are actually developed to deliver them. So that needs to be understood. Next slide, please. So the choices of the technology, this is where the instructors and institutions, they need to um, take a proactive role and also guide the students what is good for them and what is not good for them, because technology is just an instrument that can be used in teaching and learning. Technology is not everything. That's what I believe. So which technologies are you using, and what strengths and challenges do they actually present for online or blended delivery, and also for the assessment, student interaction, and student support? Sometimes we may have to use multiple technologies in order to cater all these needs. And what technologies do students use to undertake research work, for the practical work or lab work and other activities, and over and above the technologies provided by the college or universities? So sometimes the instructors need to keep an eye on the students, what kind of technologies they are using. Sometimes the technologies, they may actually put the students in a wrong path. So instructors may actually need to look at those choice of technologies for the students. Next slide, please. That's the end. So I would like to conclude that the the teaching and learning pedagogy um, during pandemic and beyond the pandemic, for example, um, is going to show a lot of influence in education sector, and many institutions are actually taking an opportunity from the COVID in order to design their new curriculums, new assessment practices, etc., in order to um, leverage the business in one way, and also to get the best of that online learning environment. So if you have any questions, I'm happy to answer.
Yes, sir. We have received some question on chat box, sir. Please.
Where can I see the chat? This one here. What is
Yes,
the
sir. It is
responsibility of
yes.
the responsibility of educational management to improve the teaching and learning process? Um, so what I would say is every educational institution, they have their own set of teaching and learning policies, and they have to abide to those policies in terms of development of any teaching and learning pedagogies. But if within the policy, if a faculty member wants to um, do something differently that would actually provide better results, I think the educational institutions will really support for that.
Thank you so much, sir, for this informative session on pedagogy development, sir. Now I would request our beloved principal, sir, Dr. V. V. Ravi Kumar, sir, to kindly present a certificate of appreciation to Professor Dr. Srinivas Nami sir as a token of gratitude and appreciation from Roland Institute of Pharmaceutical Sciences, Varambur.
Thank you, Dr. Nammi, for providing important and valuable information about teaching and learning practices. Definitely, with the advent of COVID-19, the quality of online pedagogy improved worldwide. That is perhaps the only good thing that the COVID has done to the society. Once again, thank you for accepting our invitation. In spite of your busy schedule, you spared your valuable time for us to share this important information. Thank you. On behalf of Roland Institute of Pharmaceutical Sciences, please accept this certificate of appreciation.
My pleasure.
Yep,
Thank you. Thank you very much, Dr. Ravikumar. It's my pleasure to be associated with you this evening.
Thank you, sir.
Thank you,
Dr.
Dr.
Biswamohan Sahu actually is the real coordinator for this program.
It's my pleasure, sir. Thank you, sir.
Hello, Dr. Bishwa. How are you?
Thank you, sir. Fine, sir. Thank you for your nice presentation, sir, regarding our recent teaching methodologies.
Yes, I haven't got any chance to prepare the slides, so I asked my PhD student to prepare the
Yes,
slides,
yes.
and he provided
But
those
it,
slides to me this evening.
but it was very interesting, sir.
Actually, we would
Sorry?
love to hear a pharmacology topic from you.
Yeah,
We can spend
definitely. I
the month
can.
of March or April.
I can do a pharmacology research topic, or maybe some of my online pharmacology simulations. For example, I can actually demonstrate that for your students.
So we hope that we can do in our next series. I'll be in touch with
Hopefully,
you.
yes. Yeah, no problem. Yes. So usually at the beginning of the semester, like I said, we have some busy tasks to complete. But once we start the semester, I think it's same everywhere. Once the semester starts, we have a bit of time to focus on such kind of activities.
Okay. Okay, then. Thank you, Dr. Nami. Wonderful.
Okay, thank you. So thank you very much.
Bye.
Thanks for your time. Bye-bye.
Dear participants, tomorrow we have session 1, scientific session 1 at 10:30 a.m. So please join at 10:30 a.m., sir.